Good morning, students. Today, in today's class, we are going to see about chromosome, its morphological structure, in which we are going to see the various structural parts of chromosome. So, first introduce what is meant by chromosome. How chromosome it has been described morphologically. So, chromosomes, these are darkly stained bodies seen during metaphase stage of mitosis. When cells they are stained with basic dyes and observed under microscope. So chromosome is nothing but one type of dark body which is observed during the metaphase stage. Metaphase stage is the stage at which the chromosomes they are highly condensed. So during this stage we are observing one of the darkly stained body in the cell, and this darkly stained body we call it as a chromosome. When we are staining the cells with basic dye and observing them under microscope. Chromosome chemically they are mainly composed of DNA and proteins. It is of two different types of protein they contain, that is one histone and second non-histone, with which they are highly complex structure, highly condensed structure. Term chromosome it was derived from Greek word chroma means color and the soma means body. You can see here the structure of the chromosome. Okay, so this chromosome is word derived mainly from chroma means color and the soma means body. Both these are Greek words. Chromosomes they were first discovered by Strasburger in 1875, and the term chromosome was coined later in 1888 by Waldier. So now we have introduced what is meant by chromosome. So let's look what is the actual position of chromosome within the cell. Chromosome, we know the cell as a basic structural and functional unit of life. In this structural and functional unit of life, in case of eukaryote, they have a well developed nucleus. So nucleus is the region within the cell. Nucleus is the organelle within the cell in which these chromosomes they are having their position. So they are located within the nucleus of chromosome, where they are highly coiled structure formed due to the DNA, which is coiled structure along with the various histone and non-histone protein to form this highly condensed structure that we know as a chromosome. So in overall, the relative position of chromosome within cell is the nucleus of the cell. Okay, let's see what are the different features of eukaryotic chromosome. Chromosome bear genes and thus are concerned with the transmission of character from generation to generation. So because chromosome they are having the genes which are responsible for trait inheritance, and therefore they are mainly concerned with transmission of character from generation to generation. Chromosomes of eukaryote, as we have seen in last slide, chromosomes of eukaryote they are enclosed by nuclear membrane, while in case of prokaryote they remain without such envelope and found freely in the cytoplasm. Third, chromosomes they are varying in their shapes. They are found various shapes in sizes, and they are also various in number in different species of plants and animals. So according to the different species of plant and animal, they differ in their shape, size as well as number. Commonly each diploid species contain 2n number of chromosomes. So they contain two sets of chromosomes, out of which one set coming from male parent and another set coming from female parent. Through this they are gametes. Next one, the chromosome they have the properties like self-duplication. As we know, before the start of mitosis, chromosome itself get replicated to form its identical copy, and therefore they have a property that is called self-duplication. Apart from that, chromosome have property that is segregation. That means they get segregated from each other during cell division. Apart from that, also chromosomes show mutations like various chromosomal aberrations. Chromosomes they are mainly composed of DNA, some amount of RNAs which are transcribe regions within the chromosomes, and histone protein. So DNA we call it as a major genetic part of the chromosome, which is highly condensed with histone molecule and complex to form a chromosome. Each metaphase chromosome, as we know, the metaphase is the stage of cell division at which the chromosomes are highly condensed and they are clearly visible as a darkly stained body when stained with basic dye. So each such metaphase chromosome have various parts like centromere, chromatid, secondary constriction, which is also known as satellite, telomeres, chromomeres, chromonema, matrix, and pellicle. So these are the various parts we can found in the morphological structure of metaphase chromosome. Okay, now we are going to study individually the various parts of the chromosome. So first, coming to the first part, that is called chromatid. So let's introduce what is meant by chromatid. As we know, each metaphase chromosome appears to be longitudinally divided into two equal halves. If we divide longitudinally, it is found there are two equal halves, and these equal halves we call as a chromatid. Each of these we call as a chromatid. So each metachromosome appears longitudinally divided into two identical parts, and each of which we call as a chromatid. The chromatid of each metaphase chromosome, they are having to be joined together at a particular point, that is called centromere. Okay, this particular point at which two chromatids they are found to be joined together is called centromere. The two chromatids of each metaphase chromosome, these are called sister chromatids. Two chromatids of same chromosome, they are called sister chromatids, while chromatids of homologous chromosome, they are called non-sister chromatids. Clear? Chromatids of homologous chromosome, they are called non-sister chromatids. Homologous chromosome means the chromosome which are derived from two different parents, but they contain identical genes, but their allelic content may be similar or may be dissimilar. So such chromosome we know as a homologous chromosome. And when we consider the chromatids of homologous chromosome, they are non-sister. The second part of chromosome, that is called centromere. It is important region in the morphological structure of chromosome. It is region where two sister chromatids they are found to be joined together. So the region where two sister chromatids of each metaphase chromosome appear to be joined together is called centromere. Centromere we also known as the primary constriction or narrow region within the chromosome. The main role of centromere in the chromosome, that is, it helps in the movement of chromosome during anaphase of cell division. So any chromosome, if it lacks the centromere, then it cannot be get moved during the anaphase stage of cell division. So centromere have such important role in the cell division, where they causes the movement of chromosome during the anaphase stage, at which the spindle fiber they are going to bind. At this centromeric region during the cell division, a special protein complex called kinetochore is organized. Special protein that is called kinetochore is get organized at the centromeric region, and these kinetochoric proteins are the protein on which the spindle fiber they bind to attach to the chromosome to cause the chromosome movement or chromosome separation during anaphase stage of cell division. So centromere has important role in cell division or chromosome maintenance. Based on the position of this centromere, we classify the chromosome into various types such as metacentric, submetacentric, acrocentric, and telocentric. So these are the various types of chromosome based on the position of centromere. For example, here we can clearly observe the various types of chromosome based on their position of centromere. Whenever the centromere is located exactly in the center of the chromosome, then such chromosome we call submetacentric chromosome, in which both these arms are similar. P arm and Q arm they are similar. Second, submetacentric, when centromere is located some distance away from the central position at one side, then it is called metacentric it is called submetacentric chromosome. Third one, the acrocentric chromosome. When centromere is located close to one end, then it is called acrocentric chromosome. The chromosome in which the centromere is located at one end of the chromosome, then it is called telocentric chromosome. So like this, we have different types of chromosome based on the position of centromere. When it is exactly at the center, it is called metacentric. When it is some distance away from the central position, it is called submetacentric. When it is located close to one end, it is called acrocentric, and when it is at one end, it is called telocentric. So like this, various types of chromosome, they are based on the position of centromere. Okay, the next important part, that is called secondary constriction. It is a constricted or the narrow region other than that of the centromere occurs or found within the chromosome. Okay, so secondary constriction is one type of narrow region other than centromere which is located in the chromosome structure. The chromosome which is having this secondary constriction is called satellite chromosomes or SAT chromosomes. In case of position of the satellite chromosome, some chromosome they possess in one arm, while some chromosome they are possessing satellites in both arm of it. The secondary constitution we also known as having the important role, and therefore we also know them as a nucleolus organizer region. They have a constant position within the chromosome structure, and therefore they can be used as one of the useful marker to identify chromosome. The next important part, that is called telomere, the terminal region of the chromosome on either side. Terminal region of the chromosome on either side we called as a telomere. Each chromosome it has two telomeres. Each chromosome it has two telomeres. The telomere of one chromosome, if it is break down, it cannot unite with the other chromosome because due to the polarity of effect of chromosome A, and therefore telomeres they are very specific in their sequences. They contain highly repetitive DNA sequences. Telomeres they provide stability to the chromosome ends. Okay, so maintenance of chromosome structure stable is the main role of telomere. Okay, coming to the next structure of individual chromatid, or we will observe one of the important structure, that is called chromonema. Chromonema is a long thread-like structure. Here you can see in the diagram. This is a long thread-like structure we observe under the light microscope in chromosome or chromosome arc or chromatid. Okay, and this thread-like structure we call chromonema. The term chromonema was coined by Vejdovsky in 1912. Okay, and the chromonema is actually a genetic material. It has three main functions, that is, it controls the size of chromosome, it results in the duplication of chromosome, and it is also an important portion, that is called gene bearing portion of chromosome. So chromonema is the actual hereditary material present within the chromosome structure. Apart from the chromonema, the chromosomes they also contain, that is, matrix. Okay, a mass of a chromatic material in which this chromonema is found to be embedded is called matrix of the chromosome. Okay, this matrix is surrounded by a covering, outer covering, that we call as a pellicle. So pellicle is the outermost covering or the sheet of the chromosome. It encloses the matrix as well as the chromonema of the chromosome. So both matrix and pellicle, these are called non-genetic material. Okay, so what is genetic material present in chromosome? The chromonema, a thread-like structure. Okay, and here you can see the small dot, that is called bead-like structure. So these beads are called chromomeres, which are folded structure of DNA strands. Okay, so today I hope you have understood the structure of chromosome along with its various morphological part and the relative position of chromosome within the cell. If you have any doubts, you can all of you can comment me in the comment box, or you can suggest me some comments. Thank you.